Recording. All right, we are recording. So welcome everyone to our Atlanta Affiliate Educational Webinar Series. We are focused today on pancreatic cancer clinical trials. We are so honored to have our special guest here today, which I'll introduce in just a moment. So before we get started, I do just want to remind everyone, the usual, please try to keep yourselves muted unless you have a question at the end. And please do save all of your questions for the end. We have a wonderful presentation. We want to keep things on track and your question might be answered, but then please do write down some questions that you have so you can take advantage of our wonderful guest. There also, you can put your question in the chat. We will have Barbara, if you don't mind managing that, monitoring the chat there. There is a little picture here where you can find that chat icon and type out your questions, and we'll get to it at the end. Well, welcome. We are so excited to have Dr. Loaiza-Bonilla here with us today, the Medical Director of Research at the Cancer Treatment Centers of America. So without further ado, I'm actually going to stop sharing my screen and let him do his thing.
Wonderful. So nice to see everyone. So I think I just need to get access to like the share screening capabilities, and that's it.
Okay. With that. Okay, just a moment, guys.
Yeah.
All right.
Okay, so well, nice to meet everyone, and thank you for the opportunity today to present. Dr. Arturo Loaiza-Bonilla. I'm practicing medical oncologist. I'm the Enterprise Director of Research and GI Program Director at Cancer Treatment Centers of America. I am passionate about research and genomic testing and finding options for my patients, and pancreatic cancer being one of those that really piqued my my interests since my early years in fellowship now several years ago. So very happy to be here and answering questions from anyone who's interested. So today's meeting is going to be about clinical trials in pancreatic cancer. What are the options that currently we have available beyond the standard of care? What is exciting? What's new? What do we have at CTCA and beyond? And also trying to find what are the key challenges for clinical trial enrollment? What are the solutions that we can use to help patients get access to those agents and those studies, and kind of opening a conversation for that purpose. So without further ado, so there you go. You can follow me on Twitter as well at Dr. Bonilla Onc. We have very interesting conversations there for many topics, but typically we have a Tumor Board Tuesday where we discuss GI oncology research in different cases. So you want to be just listening to a bunch of folks from the whole United States and beyond discussing cases about GI oncology. Happy to join. I'll have my disclosures here. So today's topics, we're going to talk about the latest and greatest. Of course, I'm always exaggerating, but anything that is new, exciting, something that we want to be looking forward to in terms of research. Then we're going to talk about why patients should consider clinical trials, and what studies do we have open in Atlanta and also other hospitals at CTCA, and why clinical trials are important not only for pancreatic cancer patients, but patients as a whole, particularly patients who are underrepresented in clinical trials. So this is all about pancreatic cancer. Of course, it's a PanCan one, but once again, this is one of those diseases that are really hard to manage, and we want to know why. So many of you know this already, but it's good to go to a quick reminder. So first, the natural history of pancreatic cancer is characterized by changes at the DNA level that contribute to the formation of it, and also lead to resistance to therapy, progression of disease, and we need to understand those mechanisms so we can fight them appropriately. Most pancreatic cancers arise due to multiple mistakes that happen at the DNA level. And some others can happen because of genetic predispositions, such as BRCA gene mutations and whatnot, but many of them are just a number of mistakes that happen at the DNA level. There are some rare subsets of pancreatic cancers that arise in association with a genetic alteration that are targetable, and that's something that we want to discuss because that's a personalized approach that we can do in your particular cancer. The stroma, which is basically all the the cells, but there's also a microenvironment. We live ourselves here, but there's a building where we live, right? We have where it's made, of walls and windows. The same happens around the cancer cells. So all the connective tissue around it is called the stroma. And that stroma becomes like a cocoon, like a bunch of fibers that are made by special cells called stellate cells that are inside of like accompanying the pancreatic tissue. And those stellate cells make a lot of scar tissue around their fibroblasts. They're specialized. Sometimes they have vitamin D receptors, so people are trying to attack those ones to see if we can decrease that amount of fibers around. But imagine if you clutter a place, it makes it almost impossible for things like chemotherapy to penetrate in those tumors themselves, and also for immune systems to attack. If the police is trying to get to a place that is fully cluttered and flooded and crazy, then there's no way that it's going to be able to find the cancer and attack it. So that's something that we want to attack as well, which is that stroma. And lastly, we also know that there's different genomic alterations or subgroups that we can help to differentiate, so we can give them appropriate treatments and a tailored approach. So that's all the background for the clinical trials. There is a paradigm shift, which we call, in the past, we used to call things based on the origin, you say pancreatic cancer, lung cancer, etc. But we realized then that it's not only about appearances. You can look at the tissue and look, oh, it looks like a pancreatic cancer, but at the same time, you have to look at the biomarkers at the DNA level, at the proteins that are expressed and all that stuff, because that leads to potential opportunities for treatment that we can potentially exploit. There's a number of ways to do this. So the first thing is something called immunohistochemistry, which is basically looking at external proteins that are expressed. So we use special stains, the same way we recognize the cancer type by looking at different stains that, based on a sequence of them, we can tell this is pancreas cancer versus a neuroendocrine tumor, which is also in the pancreas. They'll have different features. Then we can differentiate by immunohistochemistry. At the same time, there are certain biomarkers that we can also detect by immunohistochemistry that can help us, do targeted approaches. The other techniques are called FISH, which are, for example, to look for things like HER2 positivity and changes at the DNA level. So we can see, in this little graph, the changes in color. So when there's a change in the gene, so one gene is yellow and the other one is red and the other one is kind of like blue. So when you combine it, it shows the green color. That's when you start seeing those FISH changes. So that's something that you can tell. We look at fluorescent light for this. And lastly, is called next generation sequencing, which is basically, the DNA is a book, and the book has the instruction to make, a recipe, whatever you wanna make. But there's typos in that book that eventually, when there's so many mistakes, the cake comes horrible and it tastes terrible. That's what happens at the changes at the DNA level. And if we're able to use a technique that reads all the pages and tells me, okay, there's a typo, instead of putting salt, it says sugar on page 23 on the key lime pie. And there's another one in page 82 where there's in specific paragraph 2, there is a change here as well that makes the recipe taste horrible. Then we can potentially correct those mistakes somehow by finding targeted drugs that go specifically to stop that gene that is driving the cancer growth. And that's done by reading the whole part of the genome that is affected. That's called next generation sequencing. So we can do it in 2 ways, that next generation sequencing, looking for many biomarkers instead of just doing one. And that could be done by tissue-based assays, which is basically taking a piece of the cancer, looking at the DNA level, sending it for readout and do that process. The other one that is coming now, is becoming very relevant, is liquid biopsies, basically drawing blood from you, like two vials of blood, the same as they get your CBC or your normal chemistry. And in about like a week or something, you can get results equivalent in terms of finding those biomarkers compared to tissue, which sometimes takes longer. So they're both are very helpful. But when sometimes it's hard to get a new biopsy or find the tissue, particularly when the pancreas is done by fine needle aspiration, then this may be helpful for finding potential options for your care. So we know that in cancer as a whole, there's a lot of different biomarkers. So as you can see in this list here, it's not comprehensive, but it's big enough that it tells you a number of biomarkers that we can target. Some of them are shared between different GI malignancies, as you can see here. But in pancreas, we started to look for things like MSI status or TMB. Now, KRAS, which was an undruggable drug, now is becoming to the forefront. Actually, as we speak, the ESMO meeting, which is the European Society of Oncology and Hematology meeting, is happening, and we're getting new results, particularly in colorectal cancer, that potentially can be extrapolated or used for future research for pancreas cancer. So I'm excited about that for sure. So why is this important? Because we already know what these cancers are made of. So when you look at different subgroups of patients with pancreas cancer, they give you, on the right-hand side, you see all the genes, the names here, KRAS, P53. So you see pancreas cancer has different subgroups, and you can classify them by these 4 subtypes: stable, locally arranged, scatter, and unstable. And they're just basically based on a combination of these, genes that are altered. And you can see frequencies. Most of these cancers have KRAS mutations, but not all KRAS mutations are the same. They have different subtypes or variants. So the same as, Jane. Yeah, Jane who? Smith, Jane Martinez. Is she Latino? Is she? So you don't know, right? So you need to know basically the variant that is specific for your cancer, but it can be as frequent as 95% mutations. There's also P53, SMAD4, the well-known BRCA1 and 2, which are predisposing for other cancers, such as breast cancer, and we are targeting now ovarian cancer. So when you go deeper, there's an article from Nature where it just tells you, even looking at the RNA level, at the signaling in terms of how these cells behave, there's a lot of things that are happening that are mediated by these genes that are altered, as you can see here. And they can lead, some of them may be coming because of pancreatitis, some are because of DNA damage repair, problems with the cell cycle. So all of them are subtypes of cancers within the pancreas itself that we can potentially attack. So that's why no cancer is the same, no patient is the same, everyone is different, and that's how we do personalized medicine, right? So one broad approach is helpful, but is not ideal when we're looking at pancreas cancer particularly. Why? Because we know, for example, there's some patients who have, the BRCA mutations that we give them platinum-based agents and they tend to respond very well. So you say, oh, I got, Gemcis, or I got FOLFIRINOX or FOLFOX, and I did great and I got a response, and the neighbor and across the, the chemo chair say, oh, I got that and I did horrible. It didn't help me at all. But I got, gem and nab-paclitaxel that helped me. It's like, oh, it didn't help me. So it's because there are all these different changes at the genome level that we can certainly look into to understand the responses. For those patients who have BRCA mutations, there's already a drug approved, which is, this is the POLO trial that was published about 2 years ago. This is Jose Baselga, rest in peace, but he helped us to advance a lot of the science in breast cancer and also in pancreas cancer. And this was basically using patients who got platinum-based treatment, typically, FOLFIRINOX in first line, and got a BRCA mutation. They were able to be on maintenance with this pill called olaparib, and right now it's an option for certain patients. So always look for 2 kinds of testing: germline testing, which is looking at your genetic predisposition to have cancer, and also it may help you to genetic counseling your family, and genomic testing, which is looking at the DNA of your cancer. So they're completely separate tests, but they're both helpful for you to understand the different options that you may face. There's rare biomarkers that also appear in pancreas cancer. One of them is these NTRK fusions. They are driving cancer growth, and if you find them, is like getting the jackpot, quote unquote. Once you have found that you have cancer, you need to get some good news. If you find these alterations, you may be one of those. So those patients who have these NTRK fusions in their cancers, they tend to respond to these drugs that are oral drugs taken by mouth. The other one is tumor mutational burden. So I was mentioning that we do next generation sequencing, and we're reading the whole book. So the whole book, when you read it out from the DNA, then it gives you a number and tells you how many typos per page of the book are in the cancer. So in this case, how many mutations per megabase? That's how we call tumor mutational burden. Typically, pancreatic cancer tends to have a low TMB, that's how we call it, but there's some patients that actually respond well to immunotherapy when they have more mutations than normal. Why? Because when you awaken the immune system with immunotherapy, you are seeing a cancer that is making a lot of mistakes. It's very sloppy, has a lot of errors happening in the DNA, and that looks like a lot of garbage on the bloodstream. And the immune system just catches it and goes and finds the source and tries to kill it. So different cancers have different thresholds for tumor mutational burden. The FDA has approved pembrolizumab for TMB 10 and above. So just be always on the lookout for your TMB, because it may be rare, but you may be one of those. I have a number of patients. So I'm just going to give you a story of this. So a 66-year-old male, he presented to see me with pancreatic cancer. He initially had a cold and coughing 2 years prior. They found it was pneumonia. It wasn't COVID, it was before that. But he was presented with hemoptysis, was treated at a VA hospital, transferred to see a primary care doctor. They did a bunch of scans, found a bunch of lesions on his lungs, and also multiple liver lesions, some of them as large as 2.4 by 2.5 centimeters, so basically an inch and above. They did a liver biopsy and also pancreatic, an ERCP, because there was a biliary obstruction. They placed a stent, which is many of you may have faced that situation, and it was referred to CTCA to CS. This is how, if you haven't seen this before, this is how the needle form here. You can see the shape of it, and it contains the pancreas tissue, which looks very abnormal. Has all these odd cells kind of piling out. They're not organized, so you knew that it's abnormal. They did a biopsy of the liver, which also confirmed that there was cancer involved in the liver itself. When you look at deeper, they look at these very abnormal looking cells. They're not like round and nice. They're just like kind of messy and big. The nuclei is kind of different stages. That means the cells are dividing actively. And they did a special test by staining called PD-L1, which typically predicts response to immunotherapy, not typically in pancreatic cancer, but in other cancer types. So something that they just wanted to do to see if there was any opportunity for immunotherapy, and the answer is probably not based on this. So got the RCP, all these things happened. Finally, we solved the bilirubin, which was the major issue, but then we sent for genomic testing, right? So, and this is what it came up with. So it came with a tumor mutational burden of 66 mutations per megabase, which is very, very high. As I mentioned, typically is in the like 1 through 4 mutations per megabase. So this is pretty high. Other mutations as well that we can potentially target in the future, but basically this patient was one of those that we say, let's try immunotherapy for them. These are the options, all these immunotherapies could be used, but mostly pembrolizumab was approved for that patient. So starting pembrolizumab at the time was off label, but the patient was in a clinical trial later, and that's how we got the approval for these drugs now, because now we prove through clinical trials that those patients do better than patients who are treated with conventional chemotherapy and had a good response. So this is just a before and after. The patient is ongoing and doing okay. So basically, this is the initial scan with all these, round lesions here. This is all the liver. This is the patient lying on the table. This is the back. This is the bone here, the spine. This is the front, the belly. This is the right side, which is the R, and the left. This is before and after. So you see, this is the liver here. It looks very heterogeneous. It doesn't look nice. It just has all these like black holes here. This is all cancer. After a few cycles of pembrolizumab, you can start seeing this is clearing, right? Just a few things here and there. They put a needle on these spots that look abnormal, and actually there was a bunch of lymphocytes, basically the immune system just going there and attacking, but they couldn't see cancer. So, so this was like a great response, and patients like this are the ones that, make us wake up every day trying to do everything for every single patient. So this is the good things for us we want to do. Okay, so what are the new things happening beyond what I mentioned? There is certain biomarkers that are also done by immunohistochemistry. One of them is claudin 18.2. It's also expressed in pancreas cancer. There's a CAR T cell now that is actually just being tested for these drugs. So very interesting. We just saw an ESMO yesterday. And there's also these drugs in development with the zolbetuximab that we're waiting for the readout of the phase 3 clinical trial. So very exciting to learn about these ones too, because really helping us to fight the cancer better. We're following these patients closely with these liquid biopsies. So they're not only helpful for detection of targetable mutations and finding treatment options, but also helps us to find if the patient has still cancer or detected before it recurs and becomes evident on the CAT scan. So there's two ways to do this. One is taking a piece of the tissue and sending for testing, which is called the tumor-informed approach. And then the left-hand side is the one called the epigenomic and fragmentomics assay, which is basically just drawing blood, sending it out, and getting those things. This is the Garden response, and this is the Signatera MRD testing. So for you to know, those are the two that are available. I already discussed this. Okay, so other interesting results that we're waiting to be reading out. So this is the Combat Keynote 202 trial. This is a brand new trial that we're waiting for expansion. But this is the phase 2 results that were reported just recently of this new drug called motixafortide. It's kind of weird. Everything becomes like alphabet soup these days. But basically, it's a drug that is used for a stem cell mobilization for multiple myeloma patients. So nothing to do with pancreas cancer in the past. So basically, it's a drug that is used to when you're getting a transplant and you want to harvest your own cells before you drop chemo, and you can do your auto transplant. They use this to release the stem cells from the bloodstream and from the bone marrow and recruit them. So they're trying to see that it seems like this drug as well has some involvement on the microenvironment, basically making the immune system cells more receptive to giving them a signal with immune checkpoint inhibitors, like with immunotherapy. So basically making the police being, hey, the general is calling you, use this ID, you can actually go and do what you're supposed to do. So they're trying to combine it with chemo, and we're seeing interesting results that hopefully we can replicate further. But you can see here, for example, disease control rate was 63.2%. This other patient was already pretreated. So we're looking to see what's happening. So hopefully we learn more soon. There's other drug called the Mivystat as well. This is from the Avenger 500 trial. Basically, this is FOLFIRINOX plus the Mivystat versus FOLFIRINOX alone in patients with stage 4 pancreatic cancer, and they go from 18 to 75 years of age. It's a phase 3 clinical trial that just accrued, so we're waiting for the results. The only thing we have right now is the phase 1 results that basically showed an overall response rate of 61%, which basically that 61% of the patients had some form of response or stability on their cancers. And they had a median overall survival of 19.9 months, which we had not seen before. I think we really want to get these results out soon. So hopefully we will learn more about this study. So exciting things happening. So for those who don't know, what are clinical trials? You spoke at length about it. So what are they? So basically the research studies, this is the definition from PanCan. So I'm just stealing that from them. So that investigate new treatments or new combinations of treatment. You want to get all these studies being done so you can get approval by the FDA and get access to those drugs right now. So in the fight of pancreatic cancer, we recommend clinical trials because that's the best way for you to access those treatment options right now, and also helping to know the contribution of science in not only yourself, but many others in the future. These patients who have responses are paving the way for others as well. So it's, and not only that, but you get a lot of help on top of that by doing clinical trials. There's an exciting initiative by PanCan called the PanCan Precision Promise Adaptive Clinical Trial that is, nationwide. So feel free to go online and check PanCan resources. This is a very interesting portfolio of studies that can really help us to, find better options for our patients. So why you should consider clinical trials? First, these are the safest environments for testing new drugs because you not only have the FDA on top of you, you have the CRO on top of you, you have the sponsor who wants to get approved, you have the investigator invested on this because the reputation is on the line, you have a research coordinator who goes to the excruciating minutia to make sure that the PI is doing the right thing and makes them sign every single form possible. So I can guarantee you that we are on top of patients beyond standard of care. So it's always a good option. Participants in clinical trials can get access to drugs earlier than most folks. And in cancer, we know that many patients are actually getting a standard of care plus. So the bare minimum on the bar is the standard of care. the clinical trial is just a chance to get the extra drug on top of it. So you're not losing anything, and you may gain additional options for your care. Of course, the trade-off is sometimes you have to get more phone calls or being more often in clinic. So everything has to be discussed, but just something to always be mindful of, because we should all be looking for clinical trials. In terms of CTCA, we have a way to find those clinical trials as well. So cancercenter.com is /clinical-trials, where we have all those clinical trials. You can search and filter and schedule whatever you need to, so we can help you out as much as possible. We also have some resources. So this is a resource that I wrote as well with our Vice President of Research, which is called Our Cancer Clinical Trial Safe. And it's a very comprehensive resource you can access. Just type Clinical Trial Safe Bonilla, and you probably find me, or Cancer Center, or CTCA, and you probably find those that paper with all this extensive information. So with all that, let's talk about what clinical trials we have available at CTCA right now. Of course, these things change. So this is a recording. That means that by the time it's there, if you're looking at this, it may not be the same. So just check back again, right? So, but this is what we have open right now as of September 2021. So we are members of NCTN, which is basically the network of affiliation with the corporate group studies and the National Cancer Institute. And because of that, we can open these studies at will once a patient becomes eligible. So these 4 studies are listed here. Basically, there is one called 8021806, which is a phase 3 trial that compares perioperative FOLFIRINOX, so before and after surgery, compared to adjuvant chemotherapy for patients with pancreas cancer has been removed. The other one is the Apollo trial, which is for patients who have the BRCA1 and 2 or PALB2 mutation and are resected, and is basically using this drug called olaparib for those patients in a form of adjuvant setting. The other one is for elderly patients. You know, elderly patients are suffering from pancreas cancer, but they may not be able to get FOLFIRINOX or something like that because it could be too rough for many patients. So the idea here is to use a combination of different drugs to test in those patients how they do. And lastly, we have one for BRCA1 and 2 mutations in maintenance, similar to the Apollo trial that we discussed as well. But in this case, we're adding pembrolizumab. So we use immunotherapy on top of the targeted drug in the S2001. So all of them are available right now. If you're interested, happy to learn more, just reach out to us. We have to discuss them. Sponsored trials, which are basically brought by industry. We also have an open. This is the Golden Biotech GH-Bank 1001. Is basically gemcitabine and nab-paclitaxel. So as you can see, a standard of care, but you also get the add-on on this drug called anthraquinone, which has anti-cancer properties and is a pill form that you take basically 3 times a day and is already on the extended phase. So you know exactly what you're getting. And already past the PK part, basically there's less visits involved. So it's much easier than when you were initially part of the phase 1 component, which is already passed. So that's open and available for any patient. Other studies that we have available at CTCA, which we're very well known for, are these large trials called, there's 2 kinds of multi-drug trials and multi-disease trials. The one is umbrella trials, which is basically looking at different drugs on a single cancer type. So there's a number of them available. The ones that we're most well for are the basket trials. The basket trials is that, you know, imagine you're in a picnic and there's a lot of baskets. And then is that, oh, from where am I going to use, you know, I want to get an apple from, well, I'm going to go to, so what is your name? My name is Andrew. Okay, Andrew, so go to letter A. That's the arm A, go onto that basket and get the drug there. So these are studies like TAPER from ASCO or Tapestry from Roche Genentech or NCI Match from the National Cancer Institute, where based on the mutation that your cancer has, or you can have multiple mutations, you choose one arm and then stick on that treatment for that period of time. So one of them is Tapestry. We have open, which is Roche Genentech, and CTCA is collaborating with that. It's a just-in-time activation, basically, that the study doesn't open until we find a patient. So it's cost-effective, it's helpful for patients, and it gets activated just when a real patient is coming our way. So the idea is to do this comprehensive genomic testing for every patient. If we have the alteration in any of these arms, and those arms are growing pretty much every couple of months, then we can put you on those arms of different options. So you can see there's one for Pancreatic kinase mutations can happen in pancreas cancer. There's AKT, NTRK fusions, ROS1, even the TMB. You see now they're enrolling for 13 and above mutations per megabase. So you can get those drugs in access as well. And there's other cohorts coming your way, including RET fusions and BRAF mutations. So very relevant things that are happening. Okay, so now that I spoke about the interesting thing about genomics, other part that I feel very interesting is how can we improve access to patients to clinical trials, right? So we need to focus on patients who have, diversity background. We want to make sure that everyone gets it. Why? Because cancer affects everyone. So 18.1 million individuals were affected by cancer in 2018, 9.6 have mortality from that, and 23.6 millions of patients, new cases every year by 2030. So, this is a burden that's not going to go away. So we want to focus on patients. So for example, Hispanic health, cancer is actually the top disease for them, beyond myself included. I'm a Hispanic, so yeah, I'm included there. My family, etc. So of course, I'll focus close to my heart. The kind of cancers, they are different for every single, background that you may have. So this is, for example, the white Caucasian versus an African American versus a Latinx or Hispanic. All of them have different prevalences of cancer. So we need to be very personalized in the way we look at this, because despite good incidence improvements across the board, there's some of the cancers that actually have been worsening, and pancreas cancer is one of them. Like, is one of those that the success story for phase 3 clinical trials has been dismal for many reasons, one of them including low accrual. So we need to make sure that we enroll in clinical trials. This is African American patients as well. You can see a little bit of a difference as well, but pancreas is very common here, in terms of lethality, in terms of frequency as well in males and females. So we want to reach out to those patients because they need to make sure that we get into the best options for them. So racial and ethnic minorities in clinical trials need to be included. This is not a new thing, and I just don't discover, the moon. Oh, there it is. We all know this is happening, but we need to come together as as medical organizations and as patient advocacy groups and society to help everyone get access, because this is going to translate into better outcomes for everyone. So the disparity is real, and we know that 12% of African American and Hispanic patients, are 16% that are in a population in the United States. But look at the proportion of participation: 5% African American and 1% Hispanic. And this is just of the piece of the pie who actually enroll in trials. Across the whole spectrum of the whole society, in the last 10 years, it's been 12 million individuals who have had cancer. Only 0.1% have got a clinical trial in first line. So this is just crazy. I mean, there's so many studies, and half of the studies close because of lack of accrual. That means is all this effort, all these initiatives, all these things that could help patients in the future, they're just sometimes lost in that, in all that chaos. So we need to improve that, and we want to make it by making a direct approach to patients. So first is trust. I mean, this is a topic that is across the board. It is not to a minority, it's actually relevant to every single patient. We need to trust our providers, and that comes from information, but getting engagement, by understanding what's happening, and we are really there for you. We dedicate our lives for patients. We had family members affected by cancer, so we are committed to help you there. So, listen to us and make your own decisions, of course, but, see us from a perspective that we're really trying to help as many patients as possible. I know there were, mistakes done in the past by other folks. We're trying to, improve that and be better individuals and better professionals. So just open up and make sure that you listen to what we're trying to tell you. Access and knowledge, right? So we need to make sure to simplify concepts. People need to understand what's being offered, not just do as I say. but understand what I'm telling you. And that's what I tell my patients. Okay, if there's anything you did not understand from this conversation, you have to ask me. Like, don't leave until you actually understand everything I tell you has to make sense. You're the boss, you make the decisions, but they have to be informed decisions. They have to be decisions made with the information that you're supposed to have, right? So the same way I treat my family or explain my family, I want to have the same for my patients. That's what we want to do every day. And lastly, social determinants of health. Many times we forget, we're all human beings, but sometimes we're privileged and we don't understand what others may have challenges getting access to things such as, transportation, parking, daycare, taking care of a loved one. We have an elderly parent that you have to take care of. All those things are key, and we need to be able to overcome those challenges to help patients to get to clinical trials. So this is from the Kaiser Foundation. So all this are personalized medicine. So you may think, oh, it's only genomics and science. No, it's all that, access to options, literacy, language, transportation, income. All of this is key so we can overcome the challenges for clinical trial enrollment. And we need to be very comprehensive in that take so we can really help everyone involved. So let's be part of a community, let's help each other, and let's use technology and scalability to do that. So that's the key thing for me. Like, there's ways to overcome this by using technology to decrease cost, to make things much easier for all of them involved. So workforce. So in addition to improving diversity in the physician workforce, which is long-term, we need to make that workforce very competent in understanding their needs. Costs, we need to decrease costs for patients, and the whole system may benefit from clinical trials actually. In access and knowledge, as I mentioned, we need to make sure that people understand that these are options for every single patient available. In every single stage, even early stage, there's a clinical trial. It's just a matter of finding it, and there's ways to do this. So if the FDA has guidance, great to have, good to read, but it has to be practical. So we're trying to do this. We can use strategies by connecting social services, addressing social needs, use community health workers, all these things as a community. Like PanCan, for example, is a community of advocates, patients, survivors, caregivers, and legacy folks who are doing it on behalf of someone that they love. All of them, they come together to really make something meaningful. We need to capitalize on those initiatives and really take it to the next level. So using social media, you have Facebook, you have Fitbit, all of that. This is driving the path, and we use it right now, video to kind of have some widespread knowledge. We can use it using apps as well. So cancer mobile apps are increasing every single day for every single type. I see only 15 for pancreas, so let's try to make it better. They can be used for wellness, for diagnosis, treatment, monitoring, clinical trial seeking, and also for wellness as a whole and pain management and things that are important. So let's try to use technology for that. So for that, I have two great options for you. So one of them is the Clinical Trial Finder by PanCan. So you can access it here. You can see, you can know all your options, either as a healthcare provider or as a cancer patient or a caregiver. It gives you everything about the clinical trials that are available, and they try to help you to get there. Also have the Synergy AI clinical trial matching solutions, an app as well, that it looks for 30,000 clinical trials, and they also help you to find the solution. So it is very synergistic with PanCan. So use them both, use all of them, call everyone, get second opinions. You need to inform yourself, right? So use everything you have at your disposal to fight this cancer. And with that, I thank you for your time. This is just to show you that this is just on the left-hand side, the signaling pathways that are involved in cancer. So we need to find every single way to fight cancers in every single signaling pathway. And that doesn't even account for the stroma or for the immune system as a whole. So a lot of things to learn. All this is learned through clinical trials and research and technology escalation. So thank you so much for the opportunity. And of course, I open the field for questions.
Thank you so much, Dr. Bonilla. I've learned so much myself, and I've been in this space for a while, but it just is very evident that you have the absolute latest and greatest, and you are passionate, and you have a big heart, and it was just so comprehensive. And I think you echo a lot of the different things that we're aiming to do at PanCan as well. So it's just really amazing to be able to partner with you and continuing to partner with you in the Cancer Treatment Centers of America to fight this together. So again, just thank you, and certainly hope that people have some questions. And if you do, go ahead and just unmute yourselves.
Dr. Bonilla, thank you so much for sharing all this information. It is incredibly helpful, so I hope everybody on our call enjoyed it tonight. You spoke a little bit just in terms of, in particular, ethnic minorities not being involved in clinical trials for a variety of reasons, either social determinants of health or things in their environment, all of those different things. But one of the things that often happens is a lack of information. And people being afraid of clinical trials and or their healthcare provider not recommending clinical trials. So can you discuss a little bit, like, what are some of the objections you often hear directly from patients about clinical trials and how you address those?
Yeah, absolutely. One of the things that we have seen is, as you mentioned, they have no idea what a clinical trial means, right? They're like, what is that? What am I going to be a guinea pig? Am I getting the sugar pill, or am I getting the sham injection? And the answer is, we will tell you what you're going to get. So first is clinical trials, particularly in pancreatic cancer, they should be always, and it's NCTN recommended, is recommended by every single guideline, because we know that this is the way of finding better cures or better treatments for the future. A number of my patients say, well, I'm afraid of what is a clinical trial. So I had to explain to them, there's different phases, phase 1, 2, 3. In phase 1, we taste safety in the dosing. In the phase 2, we taste the safety already and like look for a signal. Phase 3, we randomize sometimes in 2 when we give you already know the dose, we're going to use that dose, and we give you the standard of care and the standard of care plus, and we compare them then to, and then we just get to approval. I also tell them that in clinical trials, you have an extra pair of eyes looking at you, because you have the research coordinator checking on you every time. So it's additional care that you can benefit from. You are getting personalized treatment in a number of options, and also that you're going to get. This is based on informed consent. So even the consent that you sign has gone through an IRB, which is an institutional review board, which is an independent review board members, including patients and advocates in the community, that actually are looking at that consent form and making sure that they fully understand what it is. They're protecting every single individual enrolling into research. And I encourage them just to read. Sometimes I just tell them, okay, let's do this. I'm going to explain you the trial. I'm going to give you the phone number of my coordinator so you can talk to her too. I mean, her, because that doesn't want to have, but him, there's also a number of research coordinators across the board. And after that, we will discuss. And there's the link for the consent form. They can read it out. They don't have to sign anything. It's a non-committal. And also, they can always stop anytime they wish. Just because you enroll in a trial doesn't mean you have to stay in that study. But of course, if you're doing well, I mean, why do you want to? I mean, it's not broken, don't fix it. That's what I mentioned, right? So if you're doing well, there's no reason for us to change. But I tell my patients, you have 3 main reasons for us to stop treatment. 1, because you tell us to, you just want to know reasons given. Okay, inform. 2, side effects are too much, so we need to adjust things or just put a pause button for a minute. Or lastly, there's not working. So the cancer is growing. What's the point, right? So as long as we make informed decisions around those 3 major components of why we're making, decisions in that, you're fine. So the studies are always non-binding. You can actually even select what you want to share on the study. If you want, the genomic testing that they did, if that's part of the study, to be stored or discarded, or I mean, you can do anything as long as within the confines of the study. So my suggestion is always listen to the options. If we recommend your clinical trial, is because we feel that you're actually eligible. There's a lot of different criteria to be eligible for. So it's not that, oh yeah, sign up and you're done. No, actually there's a lot of things behind the scenes where they have to check your tumor type, go and check under the microscope that is the right thing, checking the biomarker, making sure that your blood work aligns completely with the trial, that you don't have any concomitant medications, that you have the right line of treatment, that you haven't been exposed to certain drugs. There's all this, back end work just to get you eligible for the trial. So if we're recommending that study is because you have already, bypassed all of those hurdles and you have the opportunity to enroll. So that's what I'm saying. Don't miss that chance if you're eligible for and it makes sense to you, then I strongly consider that, because that's really the way we're finding better options for our patients.
Thank you so much. That's really helpful information. And with the recording, I think it'll be definitely good for people to hear that. So thank you.
Of course.
Who else has a question? So I'll ask a quick question. I know that you very new, as we've kind of seen at PanCan, to be able to officially talk about the blood tests. And I just didn't know if you could talk a little bit about the new blood tests and how you are feeling about them, how they're going to shape the future, and as it relates to clinical trials, doing blood tests before or after or during, and just how all that's going to work.
Yeah, so these tests are basically looking for circulating tumor DNA. So as you can imagine, cancer cells, as they die or reproduce, they release tiny pieces of circulating tumor DNA into the bloodstream. So there's always DNA circulating in the bloodstream because the normal cell turnover. So we exchange white cells, we exchange all kinds of cells in our body every single time. So there's this background of noise of germline DNA that is normal. And in the middle of it, there is the needle on the haystack, which is all pieces of DNA from the cancer that you find. This is the technique that's used now for prenatal testing, to find the biological gender of a baby when someone's pregnant, right? So or finding the abnormalities, that's the same testing. So they realize that when someone has cancer, they can find those tiny pieces of DNA into the bloodstream, and they're related to cancer specifically. And they're found at very minuscule proportions, but the technology has evolved so much that you can actually find them in levels such as, like, in a vial of blood, you can find 0.1% of circulating tumor DNA. And even finding that tiny piece is actionable. So so that's the interesting thing, that even if you find something so low frequency, you can actually do something against your cancer with that. So initially we used it for finding therapeutic options. So if someone had a mutation and we have a drug for that, then we use it. That's already established, a standard of care. We do it all the time with different cancer types, including lung cancer is the example. There's now 9 biomarkers that we can target with 9+ drugs that we can use. We want to do the same for pancreas cancer. So we got one now, which is olaparib. We want to get more and more of those. I mean, we can try NTRK as well because of NTRK fusions, use larotrectinib or entrectinib, but the list continues to expand. So the once that we found that these patients are, we can detect the DNA into the bloodstream, then people say, oh, can we use this technology to, either find the cancer before it shows up, or can we use it for monitoring to see responses? Because we have tumor markers such as CA19-9, and that biomarker typically is not as helpful because sometimes it goes up and down without any specific help. So something that we really want to emphasize is that the liquid biopsies can help us to really find options for our patients, to monitor the changes over time in circulating tumor DNA, and also to find opportunities for detection earlier in the game before it even shows up on the CAT scan.
Thank you. That's incredibly important to know and to be excited about. Any other questions that anyone has?
If I can,
yeah,
Addie, just ask one more question really
Please,
quickly,
yeah.
Dr. Bonilla. So we are an advocacy group. There's several of us on here that are representing our Atlanta affiliate, and you talked about ways that advocacy is so important in terms of clinical trials. Do you have any suggestions for us as an advocacy group in talking to other patients and really trying to get information out about clinical trials?
Yes, so I think in PanCan, we have an opportunity to really use clinical trials. Yeah, hello. Oh my God, you know, the age of Zoom. Hello, this is my daughter, Natalia. She just showed up. So can you give me a second?
Hi, Natalia. Of course. What a nice little cameo we got.
She's the star.
Yeah, my turn.
Okay, so I'm back. Yeah, these things happen. I thought I had logged my room, but I guess not.
It mixes it up a little bit. I like.
I know.
That was probably the best part of the whole webinar, Dr. Bonilla.
Yeah, she's a star. She's really the star. Cute.
Yes. Yeah, so as I was saying, for PanCan being such a big organization, you're just everywhere now with you're making the runs, having different groups across different states. I think this is the network where we can really leverage access to clinical trials. So every single new patient that comes or advocate, they should actually try to get them screened. You know, use the PanCan website and get them screened for the clinical trials. And use our network of physicians like myself. We're always happy to discuss cases and things that you need. So this is an opportunity to really say, out of 100 pancreas cancer patients that came to PanCan this week, we actually got it 100% screened for trials. So it's just a matter of having that that opportunity for them to see if they have a little report. Is that, oh, I have my clinical trial report. That's it. What I'm going to do next is up to me. But at least I know my options.
That's very important. I think, Fiona, I think to your point, right, this is something that we're really going to be talking a lot about in November and pushing, not that we don't push it all the time, but it's just that piece too of there's so many of our communities that are underserved and that are not educated and do not have the resources. So really tapping into educating ourselves on that piece and how we at PanCan and us as advocates can also search for solutions for those things. I'll do it together. November is going to be a big push for that. So this is just so timely for us to, take a look at all of the resources that you sent us. I did send the clinical trial finder for both PanCan and for Cancer Treatment Centers of America. And I know I screenshotted that wonderful article that you mentioned we could search for that really talks about, why do clinical trials and why to not be scared of them. So we'll definitely want to make sure everyone has access to that too.
Yeah, and let's have a computer active in each one of the runs. And if someone wants to know a clinical trial, then we can do a search for them on the spot. So something like that, that will be cool.
That's brilliant. Anything else anyone wants to share or ask?
I think, no, you asked all the right questions. We're always available, and hopefully this would be one of several opportunities we have discussed these things, and always happy. So engage with us. I'm available. So whatever you need, you know where to find me.
Yes. Well, thank you so much. We hope there's an opportunity that maybe we'll be able to get to see you in person. In April, we have our fundraising event, our PanCan Purple Stripe event, April 30th. So we'll be working with your wonderful team. And if there's anything that you need from us, any information that you'd be willing to share, we are here. And again, thank you so much for your time. And I just want to share what's on the screen, just talking about our Patient Central, to go there to talk about clinical trials, as most of you already know, and then to connect locally with the Atlanta affiliate. Here is the information you can mark down here to continue to connect with us and grow and have that ripple effect. So I will go ahead and stop sharing here, and we'll stop the recording. But